All right, so now that we've talked about basic Mendelian genetics, we get a chance to talk about, well, how are some inheritance patterns a little bit more complicated than what we've talked about so far with Mendel's experiments? And so on this slide, we introduce three new ways in which, again, genetics can be a little bit more complicated than one we've looked at, and we will build on some more then at the end of the chapter as well. So the three that we're going to introduce here are number one. So far, we've talked about genes that are showing what is called complete dominance, but there are some genes that will show different degrees of dominance. And so not always is a particular allele completely dominant or completely recessive. So we're going to talk about degrees of dominance. Number two, we're going to talk about when a gene has more than two alleles. So far, we've talked about for flower color, there's a purple gene and a white gene. Or for seed color, there's a yellow gene and a green gene. It's not always just two versions of that gene. You can have more than two alleles. So we'll take a look at that. And then lastly, we're going to introduce this idea of genes don't always have to just affect one phenotype. You can have one gene affecting multiple phenotypes at the same time, and this is something that's called Pliatropey. Okay, so let's start off with the first one, a discussion of degrees of dominance. So far, when we have been addressing Mendel's experiments, he has looked at characters that have genes that show complete dominance. And you can identify a gene that shows complete dominance because you will not be able to tell apart visually whether a organism is a heterozygote or a dominant homozygote, right? We talked about how those purple flowers, just by looking at the purple peas, you can't tell whether they are a heterozygote or homozygous dominant. You have to do a test cross, right? Or gene sequencing, for example. So that's an example of complete dominance. You can also find genes that show incomplete dominance. In this case, the phenotype of the F1 generation hybrids is somewhere in between the two parents that gave their genes to create that F1 generation. And so we can see this example here of snapdragons, where snapdragons, if you were to cross a red snapdragon with a white snapdragon, the F1 generation hybrids would actually be pink. So imagine just for a second, what if Mendel had decided not to use peas as his model organism, but instead chose to use snapdragons? Which of the two hypotheses of inheritance, the particulate or the blending hypothesis, would Mendel have supported with this first cross? I'm hoping that you picked blending hypothesis, right? If you take red paint and white paint, mix it together, you're going to get pink paint, which is what you get here. But again, Mendel, he would not have been satisfied there. He would have said, well, what happens next? And he would have then crossed his F1 generation hybrids to themselves. And if the blending hypothesis were true, breeding pink flowers with pink flowers, you would expect F2 generation would all be pink. That wouldn't be the case. In this case, you see that you've got one red to two pink to one white flower. So in any case, this snapdragon example here is showing incomplete dominance, where the resulting offspring from this P generation cross of a red flower to a white flower ends up being somewhere in between the two original parents. You get those pink flowers. All right, and then the other kind of dominance is codominance. When you have codominance, you have two dominant alleles that affect a phenotype in separate and distinguishable ways. We're going to take a look at an example of codominance on the next slide. But on the next slide, we're going to hit two birds with one stone. Not only are we going to look at codominance, taking a step back here, but we're also going to look at an example when a gene has more than two alleles. So maybe 3 versions, 4 versions, 5 versions. In the case that we're going to look at, we have 3 different versions of a gene. So here with this slide, this slide shows blood types in humans. We're going to be looking at codominance and then a gene having more than 2 allelic options. So knowing your blood type is important, especially in the case where you might need a blood transfusion, right? We can't give blood from one human to another human, just taking, anybody off the street, because our cells have decorations on the outside. We talked about these when we were looking at the membrane chapter, right? Carbohydrates on the surface of a cell act as uniforms, right? Identifying a self cell from a non-self cell, or different types of cells in our bodies will have different uniforms or different decorations on the outside. And so for blood types, we determine blood types also by looking at what sort of decorations do our blood cells have on the outside. They might be carbohydrates. They might also be different proteins that are expressed on the surface of our red blood cells. And there are tens of different kinds of blood types in humans, but there are two that we primarily look at. So if you know your blood type, usually when you say your blood type, for example, I'm O positive. The first part, the O, is the ABO blood group, and then the positive or negative is a different blood group. We're only going to focus on the ABO blood group first. So I'm type O. What does that mean? Let's take a look. So there is one gene in your genome that codes for your blood type in terms of the ABO blood group. And in humans, there are 3 different versions of this gene. The first version is a dominant allele, and it's a dominant type A allele. And what this allele does is it produces an enzyme that adds a certain kind of carbohydrate to the surface of your red blood cells. In this case, it's a type A carbohydrate, and in this cartoon, we're using the triangles to show that particular carbohydrate. Second allelic option for this ABO blood group gene is also a dominant allele, but this is a dominant B allele, and what it does is it creates an enzyme that will produce and add a different carbohydrate, the B carbohydrate, to the surface of your red blood cells, and we use the circle to signify that kind of carbohydrate. And then there's a third option to this gene. The third option is a recessive allele, indicated here by the lowercase I, and it does not have the ability to produce an enzyme that can add a carbohydrate, any carbohydrate, to the cell surface, and so it kind of does nothing. And so these are the 3 different allelic options for this gene in humans. Now, you get 2 copies of this gene. You get 1 copy from your mom, you get 1 copy from your dad. What you get from your mom and dad all depends on what they have to give you. If you, for example, get 2 dominant A alleles, or a dominant A and a recessive allele, you will express that enzyme that adds A carbohydrates to the surface of your red blood cells. They're going to look something like this, and you will have type A blood. The same thing goes if you inherit 2 dominant B alleles, or 1 dominant B and 1 recessive allele. All your red blood cells will do is produce that enzyme that adds the B carbohydrate. Your red blood cells will be decorated with that B carbohydrate, and you have type B blood. Next, you could potentially inherit 2 different dominant alleles. And this is where we show co-dominance. This person inherits 1 dominant A allele and 1 dominant B allele. So they produce both enzymes, the one that can make the A carbohydrate and the one that can make the B carbohydrate. And so their red blood cells are decorated with both carbohydrates, and these individuals have type AB blood. If you have type O like me, we have 2 recessive alleles. We don't have the ability to make those enzymes that can add the carbohydrates to the surface of the red blood cells. And so in terms of the ABO blood group, our red blood cells are kind of naked, and we have type O blood. So using this example, we can show that this particular gene has 3, not 2, but 3 allelic options. And it can also show codominance if you happen to inherit 2 different dominant alleles, as those individuals with the AB blood group or blood type do. Okay. And then the next deviation from Mendelian inheritance patterns is Pliatropey. So when we looked at Mendel's experiments, we were looking at one gene affecting one character, right? The color gene affects flower color. The seed color gene affects seed color, and that's it. Well, in some cases, you might have one gene that affects multiple phenotypes, and this is called Pliatropey. So in this case, here you have a chromosome. Here's a cartoon image of a single gene, and the single gene might affect trait A, trait B, and trait C all at the same time. Oftentimes, this is very apparent in genetic disorders. So in a genetic disorder, there might just be one gene that has a mutation, but that one gene with that one mutation creates a disorder that has a number of different symptoms associated with it. And each one of those symptoms is considered a character, right? So in this case, that one mutated gene can affect character A, B, C, D, E, whatever it is. Examples of this would be cystic fibrosis or sickle cell diseases.